Hello, this is AbdElRahman Elsaid. I'm going to present today a continuous Ant-based Neural Topology Search. My co-authors is Joshua Karns, Zimeng, Alexander Ororbia, and Travis Desell. As an overview, I'll talk about Ant-based Neural Topology Search, previous study, and then I'll delve into continuous ants, which is the topic of this talk today. Then I'll talk about the future research directions. So in the previous study, we proposed ants, and we used colony optimization as a basis for a neural topology search. The way we did that, we constructed a superstructure representing the neural network where each node is connected to the other nodes with edges and recurrent edges. And then the agents, the optimization agents, will swarm over this structure, and each agent will pick a path from an input to an output through the structure of the neural network or the superstructure. And then we aggregate these paths together to form generate a neural network. Then we train and evaluate this structure using backpropagation, use backpropagation for training, and then we evaluate the structure, and then compare this evaluated structure against the best performing population. If it's better than the worst in the population, we remove the worst in the population and reward the paths that the ants took on the superstructure with pheromone values, increase the pheromone values from these segments in the superstructure, and then remove the worst in the population, add the best this evaluated structure to the population, and remove the worst in the population. If it's worse than the worst, then we remove, just discard this neural network, and then we evaporate some of the pheromone on the superstructure, and then we decide if there are more of these iterations in the optimization process, and we keep this process till we reach the end of the maximum iteration dedicated for this optimization. We also designed the process to be asynchronous evolution, where we had to be using high performance computing. We had a main process that took care of generating the neural networks, and also took care of updating the pheromone values on the superstructure, and took care of updating the population of the best performing neural networks. And then the generated RNNs were sent to worker processes. These worker processes will take care of training the neural networks and evaluating them, and then sending back the results to the main process. Because the most consuming part of this process is the training and testing, if we have more of these workers, we can significantly accelerate the optimization process. The structure was, this ants had 6 heuristics. The main one was the superstructure itself, and it controls how big or small the superstructure would be. Superstructure is a discrete search space. It can be very complicated, but still it can hinder or constrain the movement of the ants or the optimization agents. So we looked at that. We wanted to get rid of this main obstacle or this main drawback. And so we proposed a continuous ant-based neural topology search, continuous ants. We replaced the discrete search space with a continuous search space. The search space is semi-continuous 3D, where we have layers representing the time lags, the movements, and the agent's movement, and on these layers are pure continuous movement. But when they move between these layers, they do that in discrete movement. So an ant would move from an input to an output by first selecting an input to move from, and then selects a layer to move on, and then it decides if it's going to do an exploration or exploitation movement. If it's an exploration movement, the ant will generate an angle theta using a normal distribution, and with the theta and its sensing radius, it will find its new location and move to that location. And if it's going to do an exploration movement, it will sense the pheromone values that was deposited and on the layer that it was moving on. It's going to move on within its sensing radius. If this exploitation movement is on the same layer, the ant will only consider the pheromone values that is in front of it, because it can't move back in time on the same layer. If it's going to move on another layer, it will consider all of the pheromone values within its sensing radius. After sensing the pheromone values, it will calculate the center of mass of these pheromone values and then move to that center of mass as its new position. The ants would keep doing that from the inputs to the outputs. An ant also would keep doing this till it reaches very close to the output nodes, and then pick one of these outputs as its final destination. And other ants will do that, and then we will collect these paths together to generate the new structure. And also the CANTS will try to condense the nodes using dbScan, so that it will, if some of these nodes are within certain proximity, it will just add them together, group them together in a single node, so it will reduce the number of nodes. We can see here on the lower hand side, or lower left hand side, a 2D projection of the paths of that the ants took. The edges that lay on the same layer are the segments of the path that lie on the same layer are the edges. The ones that are between the layers are the forward recurrent edges, and the backward recurrent edges. The backward recurrent edges are the ones that go between the layers but on the backward direction. The forward recurrent edges are the ones that are between the layers but go forward in the forward direction. This, you can see here the ants in this animation, how ants work in real time. These dots are the pheromone deposits, or the pheromone values in the search space. The thickness of these dots are how high or low are the pheromone values, and we can see that these pheromone values, some of them at least disappear as time goes by because of the evaporation. The search space, as we can see, is unbounded search space. We have 8 tunable hyperparameters in CANTS, which are have the number of in ants and exam, which is a benchmark in neural evolution. These tunable hyperparameters are the number of layers in search space, the number of agents, the agent sensing range or radius, and probability to do an exploration or exploitation movement, the condensation distance in CANTS, the pheromone update parameter, and the pheromone volatility parameter. We did the experiments using three time series data sets belonging to wind power generation called firepower plant. The parameters that we're trying to predict were, as we can see here, plotted here, are non-seasonal, acyclic, and nonlinear. So they are a challenge for a regression method. We compared the results to ants and exam on the same data set, same problem. And we saw that ants competed with ants on certain data sets and on those data sets with exam. And we also saw that ants were more concise or consistent in its performance. As we can see here, we are having a concise or a small confidence interval in these results in these box plots. We also found that not only in the performance, the error was low. I mean, with compared to CANTS and exam, we competed with them, but with the structures, the sparsity of the structures were also very competing with exam, especially exam, because exam starts its search with the minimal structure elements, unlike ants and CANTS, where it uses a superstructure or an unbounded search space. So as a summary, CANTS explores an unbounded search space. Early results competes with and outperforms ants. Hyperparameters are half of ants and exam. We also CANTS also indirectly encode the neural network topology to a 3D search space. The future directions would be to turn CANTS to a complete continuous search space. The main challenge would be to map the time lag in the neural structure to the third dimension in the 3D search space. We also willing to let the agents of the optimization process to be self-aware, to communicate with each other beyond the foraging pheromone, learn from their exploration and exploitation, learn their exploitation and exploration parameters and sensing radii from each other as they are working. We are willing to use reinforcement learning to do that. We also want to follow how myrmecologists view colonies in nature, where they consider colonies as the living organisms, organisms as the and ants as their cells. So we want to have multiple colonies that live together in parallel, and then they evolve together using a genetic algorithm, so they can change their experience and make the performance experience better, enhance the evolution experience. Last but not least, we are wanting to apply ants and CANTS to other problems with higher dimensionality. Thank you.